Welcome to this lecture on the origin of photosynthesis. Imagine being stranded on a barren, toxic alien planet with no food and no oxygen. To survive, you would need to create your own food out of thin air and sunlight. Billions of years ago, our Earth was exactly like that. Today, we will explore the incredible evolutionary leap that allowed early life to harness the sun's energy, fundamentally changing our world forever. Now that we have set the stage of a barren early Earth, let us look at how life adapted. Early microscopic life forms were struggling to survive on the very limited chemical nutrients bubbling up from the crust of the Earth. To overcome this starvation, some brilliant little cells evolved a biological solar panel. This allowed them to capture sunlight and turn it into sugary food. This incredible survival trick is what we call the origin of photosynthesis, and it is arguably the most important evolutionary leap in the history of our planet. Building on that initial survival trick, let us understand how this process evolved over time. Initially, around 3.5 billion years ago, this process was anoxygenic. This means early bacteria used molecules like hydrogen sulfide instead of water, and they did not produce oxygen as a byproduct. Over millions of years of evolution, a specific group of bacteria called cyanobacteria developed a more advanced, oxygenic form of photosynthesis. They evolved specialized protein complexes called photosystems that were powerful enough to split water molecules, releasing free oxygen gas into the environment. Understanding this shift to oxygen production leads us to a massive turning point in Earth's history. If early bacteria had never evolved the ability to photosynthesize, you and I would simply not exist today. The invention of oxygenic photosynthesis by cyanobacteria triggered the Great Oxidation Event about 2.4 billion years ago. This permanently changed the chemistry of Earth's atmosphere. This massive release of oxygen allowed for the evolution of aerobic respiration, making complex multicellular life possible, and formed the ozone layer to shield us from deadly ultraviolet radiation. To truly grasp how this planetary transformation occurred, we must look at the underlying chemistry. This fundamental chemical equation represents the transformation of inorganic matter into organic life. On the left side, we have 6 molecules of carbon dioxide from the atmosphere and 6 molecules of water. Using sunlight, these are converted into 1 molecule of glucose, a high-energy sugar, and 6 molecules of oxygen gas, which is released as waste. When the availability of carbon dioxide or water increases, the rate of glucose production generally increases, provided there is enough sunlight to drive the reaction. Now that we know the chemical equation, how do we actually find evidence of it in the ancient past? Evolutionary biologists do not just look at the equation, they look for physical evidence in ancient rocks using a process called carbon isotope fractionation. Photosynthetic enzymes have a slight preference for lighter carbon isotopes when they build sugars. By measuring the ratio of carbon-13 to carbon-12 in ancient organic matter, scientists can detect the distinct chemical signature left behind by these ancient biological solar panels billions of years ago. To quantify this biological signature, we use a specific mathematical formula. This equation calculates the isotopic signature of a carbon sample, measured in parts per thousand. The variable R sample is the ratio of carbon-13 to carbon-12 found in our ancient rock. We divide this by R standard, which is the known ratio in a standard reference material. Subtract 1 and multiply by 1,000. When the value of our sample decreases, meaning the sample has less heavy carbon-13, the resulting signature becomes more negative. A highly negative value is the smoking gun for ancient photosynthesis. Let us apply this formula to a real-world paleontological discovery. Imagine you have extracted organic carbon from a 3.5 billion-year-old rock in Australia. The laboratory tells you the carbon isotope ratio in your sample is 0.01095, and the standard reference ratio is 0.01124. We plug these numbers into our formula. First, we divide the sample ratio by the standard ratio to normalize our data, giving us 0.9742. Then we subtract 1 and multiply by 1,000. The final answer is negative 25.5 parts per thousand. Having calculated this isotopic signature, let us prove what it actually means for evolutionary biology. We know from modern observations that the signature of biological photosynthesis typically ranges from negative 20 to negative 30 parts per thousand. Our calculated value of negative 25.8 falls perfectly within this highly negative range. This mathematical proof bridges the gap between chemistry and biology. It establishes that the carbon in this rock was not just deposited by random geological processes, but was actively processed by living, metabolizing organisms. Continuing from that deduction, we can finalize our proof of ancient life. Because the value is so highly negative, it proves that the sample has significantly less heavy carbon-13 than the standard baseline. This confirms that early life was actively filtering out heavier carbon atoms, preferring the lighter ones to build their cells. Therefore, we can confidently conclude that photosynthetic bacteria were alive and active 3.5 billion years ago. This gives us a concrete window into the survival strategies of Earth's earliest inhabitants. Now that we have proven photosynthesis existed 3.5 billion years ago, we face another question. When did the oxygen-producing cyanobacteria emerge? To answer this, we use a molecular clock. This concept calculates evolutionary time based on genetic mutations. By comparing the DNA sequence of a modern cyanobacterium with its closest non-photosynthetic bacterial relative, we can count the genetic differences. Since genetic mutations accumulate at a relatively constant rate over long periods of time, trusting this steady ticking of the genetic clock allows us to peer back into deep time. To read this genetic clock, we rely on a simple but powerful formula. The time since two species shared a common ancestor, denoted by T, is equal to their genetic divergence, D, divided by 2 times the mutation rate, mu. The divergence is simply the number of mutations per base pair between the two DNA sequences. The mutation rate is how many mutations occur per base pair per billion years. If the genetic divergence increases, it means more time has passed since the two species split, resulting in a larger value for T. Let us see this molecular clock in action with a concrete example. Suppose laboratory studies show a genetic divergence of 0.08 mutations per base pair between our two bacteria. The mutation rate for this specific gene is 0.015 mutations per base pair per billion years. First, we multiply the mutation rate by 2, giving us 0.030. Finally, we divide the total genetic difference of 0.08 by this combined mutation rate. The final answer is 2.67 billion years. Now that we have calculated the time, let us understand the mathematical reasoning behind the formula, specifically why we divide by 2. When the cyanobacteria and the non-photosynthetic bacteria split from their common ancestor, they did not just take one path. They took 2 separate evolutionary paths. Both lineages have been mutating independently since that exact moment of separation. This chunk of our derivation establishes that the total genetic divergence we observe today is the sum of mutations accumulating on both branches of the evolutionary tree simultaneously. Concluding our derivation, we can see why that division is absolutely critical. If we did not multiply the mutation rate by 2, we would accidentally calculate double the actual time elapsed. This step ensures our evolutionary timeline remains perfectly calibrated. The resulting 2.67 billion years tells us exactly when oxygenic photosynthesis innovated. Beautifully, this date perfectly aligns with the geological evidence leading up to the Great Oxidation Event, showing how molecular biology and geology can independently verify the exact same evolutionary milestone. To visualize this entire evolutionary journey, let us look at this diagram of photosynthetic pathways. On the left, representing 3.5 billion years ago, we see an ancient anoxygenic bacterium. Notice how it takes in sunlight and hydrogen sulfide, releasing sulfur as a byproduct, with no oxygen involved. Following the large evolutionary arrow to the right, we arrive at 2.4 billion years ago. Here, the cyanobacterium uses evolved photosystems to take in sunlight and water, releasing oxygen gas. This simple visual captures the profound biological shift that ultimately oxygenated our entire planet. As we wrap up, let us review the key takeaways from our exploration. First, the origin of photosynthesis was a monumental breakthrough, starting as a simple anoxygenic process and evolving into complex oxygen-producing machinery. Second, this innovation by cyanobacteria triggered the Great Oxidation Event, paving the way for complex aerobic life and the protective ozone layer. Finally, by studying chemical equations, carbon isotopes, and molecular clocks, we can trace this incredible journey back billions of years. You now have a solid understanding of how early life harnessed the sun to change the world.